student who is dealing with the using of the is it black soldier?
Black soldier flag.
Yes, and I think they are continuing with that. Maybe I can only comment. You also remember that yesterday in our discussion. So what I said about avocado. So sometimes back I went to Aroko market, Kakamone here. You know, Kenya is, I think, the second biggest exporter from Africa now to Europe. But I went to Aroko market, looked to look for avocado. I didn't get them.
So it's harvesting time.
Yes, we could only get avocado from there. That is the scenario, and I think we have a book we give in our library on how to consume avocado in so many ways. I think that could help, and we will proceed doing that. But definitely we have an opportunity of promoting avocado consumption. We do eat a lot, but not like the way we eat mangoes here or oranges, because many people may not even know that avocado is a fruit sometimes, and that's how it is. So I'm very grateful to FOCLAB and our NAC partners. I think we will continue working together to expand the horizons of our research and education. Thank you.
Indeed, Eric, our mission this week is also to get an extra plus on the row of continuity. That is our mission of this week. So you're right. Thank you.
Perfect. Thank you. Any other reaction? We have one here. Please, Mr. Mike.
Thank you very much, Marco, for the good presentation. There's a presentation yesterday by one of the students who was involved in the study, Sharaf. And in one of the slides, or in the research that he did, it showed that avocado takes almost a month to get to the Netherlands. I think the Netherlands is one of the destinations that the avocados from Kenya go to. And I wanted to hear from Marco and the other researchers if there is a way that really we could also increase the consumption at home, because it really showed that by the time it reached Netherlands, a lot of that avocado almost goes to waste because of the damage during the carriage. Yeah.
Indeed, but you are right. One of the biggest problems is the damage during picking and the damage during the transport from here to the exporter who is grading. And you saw already that a lot of producers, because they were only picking A-grade avocados, but they didn't get paid 100% A-grade. There were already quite a lot of B-grade paid afterwards. And that is because of the damage during the export. So what the farmers also realized is that the transport have to be in smaller cages than just the truck, not to, and then they can reduce quite a lot of damage already. Now, here you see the picture, and this was air fried, and then they are in one day in the Netherlands. But if it is, but quite a lot of carriers, they want to have it by ship fried, sea fried, and then it costs indeed about a month. So that's true. But it is much cheaper, and so it is worthwhile. But then it has to do with the time of picking. Eric knows much more about it, so he will fix it later. Yeah, but another thing Shalev mentioned is that it seems that the dry matter content, the Kenyan avocado are known for a very low dry matter content, and you wanted to have a much. Do I say it well, Eric? You want to have a much higher dry matter content. And but we saw in this trial, because they were directly assessed, that the dry matter content was not as bad as what we always thought. So that is also interesting. So this trial also learned us that that the quality might be much better than we thought. So this trial was done just after she left the Netherlands, so we couldn't have some of those trials.
Making a conclusion that the biggest contributor of what we find in the European market, most important one, will be maturity act. And that is a big contribution from our people here. But the Kenyan government now has put in place mechanism of harvesting. So harvesting in different agroecological zones is officially open. Remember last year, so some companies were denied export license because they harvested avocado from Ghana before the opening date of harvesting. So if you found with an avocado that is going to export now, and where you harvested from is not legally opened, you risk actually being imprisoned. And there is a law now into that place. Thank you.
Thank you so much, Dr. Marco, for your presentation. There is somewhere in your presentation you mentioned that avocado is not considered as a food. Yet we know that avocados are exported to other countries for human consumption. So I wish to know the reason as to why you mentioned that avocado is not considered as a food. Thank you.
I started to mention it because on Tuesday we were talking to a former VC or DVC of Egerton University, and she was mentioning it actually. And then I thought, okay, if we had that approach from the beginning, perhaps we had done a different type of research. Yeah, so sometimes, so it is not a staple like many others. It is, as Erik said, it's not considered as fruit. If you have also a domestic market, yeah, then the export market can have a higher turnover. But now the focus is on the export, yeah, and that means that the gap in price between the export price and the domestic price is so high that it is really a pity not to go for export. Yeah, but if you farmers can get a higher income if also the price for the domestic market is going to rise. So that's also one of the solutions for them. Yeah. Now, and I think there are quite a lot of technical details we can talk about, but for me, achievement in cooperative development is perhaps the best, because here it starts. With we cooperatives, we couldn't have reached what we have reached, and they couldn't have reached what they have reached. So, yeah, and so here it starts, and with a strong cooperative, then also the achievements in the value chain can go on. So chain governance, value chain governance, for me has very much to do with cooperative governance. It's not the same, but it helps. Yeah.
So a clap for Tutagi as he sits. Wonderful.
Yes, Dr. Gunnar can hear me away as I confirm once again who is present. So do we now have Mweni Paul, who appears as the third presenter on our program? Okay, still not here. Do we have Victor Mugambi? Okay, so we proceed with the first one, of course, will be Dr. John Gunnar. Of course, John will come second. Then we will have Kimathi Aaron. Are you here? I think Kimathi was here at 6. And then one was there from Margaret's review.
Thank you so much, Dr. Gashoka. Good evening. I'm Joan Gunnar, and I'm going to present on a research about microplastics. So, to start with, have you ever wondered that the plastic bottles that we use for our waters, you see all around, the plastic bags that we use for our packaging could really affect the quality of food, or rather the plants that we consume. So this is what inspired us to look at it. Microplastics, as it is an emerging issue, environmental issue, concern globally now. And this is because of the fact that one, it is non-biodegradable, and two, of the effects that it has, it can be able to be a carrier to other pollutants like heavy metals. So heavy metals can absorb on the microplastics as a base and therefore be carried over. And because of that, then it is important globally now that we take care of the environment by ensuring that we are not offering conduits for transfer of microplastics. So microplastics generally are plastics that have disintegrated to a level of below 5 micrometers. So this is unique in such a way that you use the plastic with exposure to sunlight and other factors, it breaks down into very small particles. Actually, we are also now in the era of nanoplastics, which means that these microplastics can further degrade down into other finer particles. Now, why is microplastic a concern currently? It is a concern because it affects almost all aspects of the environment, be it in the waters, be it in the soils, and also in the biosphere, that is the plants and animals. Now, in terms of the soils, microplastics are capable of altering the soil structure, thereby affecting the bulk density, so in terms of how much the soils can be able to take. It also affects the water holding capacity. So literally what this means is when we have microplastics in the soils, then the capacity by which the soils can hold water is affected. Actually, it is lessened. Now, also, as a factor, because of this, then, there are studies that have showed that microplastic can be transferred transferred from the soils into the plants. And when the plants uptake the microplastics, then it affects their physiological structures. And by so doing, then, of course, the nutrient levels in the plants are affected. Remember, for you and me, we consume the plants because of their nutrient levels. And also, it bioaccumulates in the plant tissues, which means that when you consume part of the plant, be it the leaf, the stem, or the roots, then there are chances that these microplastics could be transferred from the plant into your body systems. And if that happens, then the detrimental effects to human beings and animals vary from respiratory issues, cardiovascular issues, neurological issues, and reproductive issues, not to mention that microplastics on their own merit are carcinogenic. So we wanted to find out, are there chances that microplastics are available in our soils? Of course, from previous studies, yes. But are there contributing factors to this? So we looked at our local environment. What do we do in our farming systems? More or less, we use inputs, and some of these inputs could be fertilizers, could be pesticides and herbicides. But for the fertilizers, for the local communities, most of them go for composted manure. So for the composted manure, this is made out of several other products. We can use the animal waste, we can use household waste, we could also use plant waste to compost this manure. Now, can this manure be a factor or a pathway of the microplastics into our plants? So this is what this study sought to find out. So from the sources into the soil, they are varied from fertilizers, from the polythenes that we use in our greenhouses, and I'm sure this is very common in our households, the greenhouses, from the plastics that we use in mulching, especially in large plantations. I'm sure around this area or on the other side of Nanyuki, we have large-scale farming going on, and more often they use mulches. We could also get it from sewage sludge. From most of our municipalities, we derive the sludge from our sewage system, and we use that as fertilizers, and of course directly from our animal waste. So when all these are combined together and we use it as composted manure, chances are that this could be transferred to our soil. So this is the hypothesis by which this study worked on. So what did we seek to find out? We wanted to know, are there microplastics and what are their levels? How much are we getting or are we transferring into our soils when we use composted manure? So we also wanted to find out, does the type of the compost manure affect the quantity? So we looked out for 3 types of composted manure, majorly from animal waste. So this is from the goats, the chicken waste, and the cow dung. And also we wanted to find out if these microplastics are there, what is their compositions? In what morphotypes do they come in? So with this in mind, we looked out for farms that have been using this composted manure, and those now became our sampling sites. So we did purposive sampling. We looked out for farms that have been using this composted manure, these 3 types, for the last 5 years. Why is this important? Is because the history of application affects the abundance. So we limited it to at least 5 years to see if by that would there be a carryover, would the accumulation be a factor. So we chose 12 sites within Embu County which had a history of this application. Now, we sampled the soils from about 15 centimeters deep into the soils. We dried the soils and homogenized it. After that, we sieved it using sieves at the level of 5 millimeter pore size. Now, this is important because remember, microplastics are anything below 5 millimeters. So after that, whatever we got, we did wet peroxide oxidation. So basically, this is digesting the organic matter with 30% hydrogen peroxide with a mix of Fenton reagent, this is basically IO2, to enable the decomposition of the organic matter. Remember, microplastics are not degradable. So anything else that is not microplastic would be digested at this process. Now, after that, the white peroxide oxidation was done several times to ensure that all organic matter that can be decomposed by the peroxide is decomposed. Then after that, we did density separation, basically because from the digestion you would sort of get some not so clear liquid, so we added sodium chloride to enable the particles to settle, whatever now we suspected was microplastic. After that, we filtered, and from our filtration, then we took it for characterization. Now, characterization was done in threefold. The first one was visualization, so using an optical microscope at a magnification of about 40, we looked at whatever we had as our residue. And for us to categorize it as microplastics, then we had to follow a criteria that by using a forceps, if we were to pick it and squeeze it, if it doesn't break, then that qualifies. So from the visualization then, it was able to give us the abundance. We were able to count how many and the shape. From the optical microscope, we could be able to tell the shape. Then thereafter, whatever we identified as microplastics, we did further analysis using FTIR. This is basically to know the type or the composition. What type of microplastic is this? So from the FTIR, then we were able to tell which is the composition, which type of microplastic do we have from the abundances. Now, in our results, remember we had three types of organic composted manure. So we had from the goat, from the chicken, and from the cow. Now, if you look at the abundances, you realize that the chicken gave the most, so that was just about 13 items per kilogram of the soil. As compared to the goat and the cow, the chicken composted, or the soils that we're using, chicken composted manure, had the highest abundance. So what would that tell you? Basically, if you look at what chicken eat, or the feeds that they use, this could be a factor when you compare it with the cow. Because from the dung composted, from the cow dung composted, that is where we got the lowest abundance. Now, in terms of the shape, now, in all, we were able to identify 4 types of shapes. Now, why is shape important? Now, the shape would almost tell you the source. The shape of the microplastic would almost tell you the source. Now, looking at our data across the 3 composted manure, we found out that fragments, averagely fragments were the largest, followed closely by the films. So if you look at the other part, basically if you look at this part, this would almost represent a fragment. So this basically tells you that this is a plastic that degraded or broke down into very small particles, and you could almost tell where this could come from. Basically from anything plastic, from our buckets, if you're using buckets, or any bottles that could be able to break and give fragments. Now, if you look at the other part, this is basically films. Now, these basically come from low-level micron-size packaging bags. Then we also have fibers. Now, fibers could more probably, in terms of the source, come from the gunny bags that we keep the feeds in, and that also can contribute to the films. And on the other side is the pellets. Now, the pellets are basically, in other words, sometimes they can be referred to as microbeads. Now, these are rounded, small rounded plastics that disintegrate. Majorly from the pellets, we get them from laundry detergents, or from packaging materials. I'm sure for most of you, if you've ever bought your bags or your shoes, normally we have some pellets, some of them referred to as silica gel. But in the essence, when these are consumed, then it would more or less be the micro beads. So if you look at it, we were able to realize that in terms of abundance, it still reflects on the fact that the chicken composted manure had the highest abundance. Now, in terms of the composition, if you look at it, then polyethylene terephthalate, which is normally known as PET, was the most abundant. And this is true because if you look at the types of material that we refer to as PET, most of them are the plastic bottles that we use in packaging of our drinks. Just one more slide. In our drinks. So this still reflected here that the most common, probable source would be the disintegration of our packaging materials. Now, comparably, if we were to compare our study with the others, then we realize that, yes, as much as we got the abundance, comparably to other studies that have been done elsewhere, our levels are still minimal as compared to others, but this could be because of several factors. Maybe because of the pristine nature of our farms and the fact that the farms were using only these as their amendments in their soils. But if you compare with what others are getting elsewhere, this is a wake-up call that with time, then we could be able to reach into this level. So what are we concluding? That yes, as much as we are using the amended, compost-amended manure to our farms, it is a risk. And in terms of which option would you go for, chicken gave us the highest abundance. And PET was the most common type. So we recommend that there is need for quality control when it comes to the use of compost manure in agriculture. Thank you.
Thank you. Okay, like we agreed, note your questions in the interest of time. So we go directly to the next presenter. And then we'll deal with all that at once. Please do that. I believe you all have a pen, a piece of paper. Okay, so let's have the next presenter. This is, can you tell us his name? But it's all in the program. I'm sure you have it ready as soon as you are.
green chemistry. I want to time myself because sometimes I get excited when I'm okay. Today I'll be presenting about an alternative environmental friendly solvent, which is supercritical carbon dioxide. When we talk about green chemistry, we talk about having invention or designing or application of these chemical products where we reduce or eliminate generation of hazardous substances. When we talk about green extraction, it's about designing extraction processes that can reduce energy consumption or allow an alternative solvent by use of either renewable natural products, which ensures safety and also good quality extract. Now, let's talk about supercritical fluids. When you look at the phase diagram, when you go at the boundary of the liquid and the gas, as you can see on the visual representation there on your left, when you start changing the temperature and pressure, as you're going through that boundary, you'll find that when initially you'll see the boundary between the gas and the liquid. But as you increase the pressure and temperature, that boundary goes disappearing until when you get to a point which is called critical point, then that boundary disappears. From there, you start another region. That region is called supercritical region. And that region, now we know very well that gas and liquids, they have very particular properties. Now, when you go to supercritical fluid, it usually have dual characteristics. It diffuses very well as a gas, and it usually have solubilities which are equivalent to those of liquids. When we are talking about extraction, we need something which will get into the matrix, so it will diffuse. Then when it gets to the unlight of interest in the matrix, then it can be able to dissolve. Gas or liquid have one of their properties, but when you are in supercritical region, then you have a solvent which have dual characteristics. It can dissolve our liquid or diffuse the gas. So it's a very good solvent. Anything can go to supercritical fluid. It all depends with what? Pressure and temperature. So if you get to appropriate temperature and pressure to a critical point, beyond there, you go to supercritical region. So all these things can go to critical points or supercritical fluid region. Like water, you are seeing the temperature very high, critical pressure very high. Therefore, as much as we know, the most greener solvent is what? Is water. But can we make it go to supercritical fluids? The condition don't allow. Because at the end of the day, we might end up denaturing our compounds when they go to extraction. Now, why CO2? Carbon dioxide have very friendly critical points, almost at room temperature, that 1 degree Celsius, and 73 atmosphere. That's around 1017 psi. It's readily available, it's not flammable and toxic, and of course, it's a byproduct of some of the processes like ethanol production. So we can be able to get it. So that's the reason why supercritical CO2 is widely now investigated or researched on when it comes to it considered as solvent. So when we utilized, for this work, we utilized supercritical carbon dioxide to do our work. The extractor is usually very simple, You have the extraction vessels where you usually put your sample, and then you have, of course, the carbon dioxide. Then you put it there, and then you can be able to regulate pressure and temperature because you have the oven there. So whenever you are doing this, there are factors to be considered. When you are doing extraction, pressure and temperature, they are very paramount. And if you have to apply this extraction, then you have, first of all, you have to do studies on the extraction part. And as you will see later, when it comes to correction, again, you have to consider the parameters, because some of the compounds are usually difficult to collect. So we applied supercritical carbon dioxide. We applied it in extraction of essential oils. We know essential oils have found their importance, especially in this era whereby people want to go to a natural, and they have found their importance in pharmaceutical, cosmetic, and food industry. So it's a big market. But the challenge which is there is that when you the solvent, which is widely used in this extraction of essential oil, is usually organic solvent. We know the dangers which come with our shortcomings. Okay, one, they are petrochemicals, and of course we know very well we are depleting that. So, can we be able to come up with a solvent which can address some of these extraction problems which come with the extraction of essential oil? Steam distillation, high heat, thermal degradation, longer times, and sometimes you end up hydrolyzing your components. Organic solvent, they are polluting organic solvents, expensive post-extraction processes, and also if it's in food industry, they usually create contamination. So there are other methods which can be used for extraction, but we really need an alternative solvent, and that's why we considered carbon dioxide. When it comes to extraction of essential oil, extraction processes, we were able to optimize pressure, temperature, extraction mode, and time. Those were the most paramount parameters to be considered. But the challenge was in the collection, essential oils are very volatile. So you might end up optimizing the extraction, but when you come to collect it, you end up losing them. So the work was done for the collection, and there were several parameters which were considered, and successfully we were able to actually extract essential oil from various plants. Then it was applied again on refining beeswax. We are talking about value addition. And sometimes we usually produce byproducts which can be used, like in production of beeswax, cappings and honeycombs are there. Both they are the ones which they contain a lot of wax. But they produce cappings and filter pits. These, as much as they are byproducts or they are waste, if you are able to utilize another method, and we utilize now the supercritical carbon dioxide, we will be able to actually extract more of the wax from those byproducts. So I want to go there, those from their studies. Then we have DDGS. These are distillers dried grains, solubles. These are usually byproducts of ethanol production. We know the technology or the research is going on, how can we reduce the usage of petrol products. Then we are coming up with ethanol. Ethanol now being produced from grains, it usually produces these byproducts. What can we do with this waste? So this waste, after analysis, it was found to have higher nutrition value, especially for those people who are diabetic. But the appearance is not very appealing. So what can we do? We can be able to extract, or we can be able to extract so that we can improve the color and also just maintain the nutrition value. And that's what we did, and we were able to actually, with supercritical carbon dioxide, we were able to improve on color, and the off flavor it had, we were able to actually also to improve on that. So we were able to actually, from the byproduct of ethanol, we were able to actually come up with a value-added product from that. So in conclusion is that we can be able to get out of from the usage of these organic solvents, and we can embrace a greener, benign solvent supercritical carbon dioxide. Thank you.
Just on time. Great. So please, once again, note the questions. We'll take them soon. So we go to the third presenter. Her name is Margaret, not Jane. No, Margaret Muriuki. 10, yeah? Yeah, on the, okay, 10 minutes. She should have presented tomorrow. She made a request, so we are taking that from the next page at the bottom. Yes, the requests. Especially now that we didn't have two of our presenters with us. Okay, we proceed.
Good afternoon. My name is Margaret Muriuki from Karatina University, the Department of Tourism and Hospitality. So I'm going to be presenting.
Microphone is on the table.
Okay. So I'm going to be presenting this research on the effects of community-based environmental governance on land conservation in the Amboseli ecosystem, Kenya. That's the bigger topic of the research. The lower one is what I'm going to be presenting on, the interrelationship between community-based conservation governance arrangements and land conservation in Amboseli. And I start with I start with the background here. Lion population trends have been on a steep decline. There was an estimate of around 1 million in 1900, though not a very good one, but it's something to go by. The most recent estimate was dropped to around 20,000 lion, and that is in 2023. And this decline has been attributed to the highest cause of decline is retaliatory killing for livestock predation. It is the most important cause of loss. And this is just an asymptomatic, like a symptom of a bigger underlying challenge, which is a challenge of governance because of a model of conservation we call protectionist, where governments, starting with the colonial governments to the current government, came up, established protected areas. Those are the national parks that we have. Then they displaced communities, and communities were left with a literal alternative to poverty, a lot of land degradation because their livestock had to concentrate in the remaining pieces of land. And therefore, there's a lot of now negative attitudes and a lot of cost to these communities because of livestock predation. And as a result of that realization that that protectionist kind of governance was a problem, community-based conservation was introduced. And it was introduced in the Amboseli in 1970s, whereby communities are involved because government had a lot of control, and it still does. So it was an attempt to involve communities in conservation, and this was started through funding from European Union and other multilateral and bilateral organizations. So basically, CBC is a hybrid form of governance involving communities, NGOs, and the government itself. So although it has been implemented, mixed results have actually been reported, and most studies that have been done actually look at the impacts of the technical impacts of interventions like compensation. Normally people are compensated when lions kill livestock. Maybe livestock enclosures, do they lead to reduction in lion killing also? There are other studies that have also been done to assess assess the governance generally of community-based conservation, but there's no study that, very few studies, let me say, that have been done to link the governance arrangements of these CBC and land conservation outcomes. And that is why this study focused in this area. And the objective of this study was to assess the influence of community-based environmental governance arrangements on land conservation, and then evaluate the variation of governance performance in the different areas of the study. So this was the conceptual framework, was derived from two frameworks. One is the governance capacity framework that is derived from a policy arrangement approach that normally has like 4 elements. That is the rules of operation, discourse, there is resources and the actors, and also the socio-ecological systems framework, that is SAFE, that has somewhat similar variables, a little bit complicated, I won't go into it, but we leveraged on the power of the 2 frameworks, because the GCF it analyzes the power asymmetry in the communities or in governance arrangements. It also analyzes the discursive, or discourse. And now the SAFE framework has a big analytical power in linking governance arrangements to ecological outcomes. And those were the study variables that were derived from the 2 frameworks. So, am I going back up?
Actually, the slides are not moving.
Okay.
On the screen.
Oh my! Sorry. I don't know what is happening. I'm moving there and, okay, sorry.
It happens. Let's see if it can be fixed. I was wondering why background has taken too long. Oh, that's better. Proceed. It's okay. You don't have to.
Oh, I don't have to go back. Okay. Yeah, so I had moved up to now I was at the study area. So this is the area where the study was done in the Amboseli region. It's in the southern Kenya. And what happens is that is the group. The study was in the community group branches surrounding the Amboseli National Park. You find that the Maasai community, they still have group branches, like a group of people, instead of the normal ownership of land where it is private, a group of people owns a large piece of land. And although that is changing because there is land subdivision happening currently, it was a kind of an arrangement that was meant to be a solution to the problem of conservation, but it didn't work. So what happens is that we have a lot of wildlife, not only within the park itself, but also within the community group branches. We have lions there. That is where they get to interact with livestock, and livestock get killed in the process. So we have a lot of interventions like conservation, a compensation of people for livestock killed. Also, there is also monitoring of lions, which is done like in a way of preventing predation. When lions are known to be in a certain area, then we have pastoralists being warned not to take their livestock there. And they have worked, and a lot of research have been done to show, yes, they do work, but there's still a lot of lion killing, because there are few studies that have really gone into an assessment of governance, which is what this study was targeting to do. So the methodology that was adopted was a mixed method, whereby qualitative data was collected and also quantitative data. And the quantitative data, we collected data from about 371 household interviewees from about 3 villages in the group branches that I've just shown, and multiple linear regression and also mediation analysis were conducted. The results were that most of the policy arrangement variables that I showed you up there, these variables, we have rules monitoring, rules compliance, financial capital, all these variables, their effect on land conservation. This land conservation variable was made from what the actors defined as land conservation, particularly community-based conservation, because it is meant to really have like two objectives, that is to conserve lions and also to advance community livelihoods or to contribute to community livelihoods. So that was a composite variable derived from all those elements. And there were some mediating variables there, which was linking capital and congruence. So most of those PAA variables there were their effect on land conservation was actually mediated by linking capital and congruence. So I will go to the data there. You can see this is the results of the regression model showing that congruence had the highest impact on, or the highest effect on conservation of lions. And just to explain congruence, or it is like governance stability from that framework that we used. It is what we call indicative governance, when their resources are adequate, rules are there and acceptable. The discourse between actors is, there is consensus between the actors, so that now that is, when it is, the stability is at a certain level, then we can say there's potential of that governance arrangement to solve societal problems. If it is too low, the stability or congruence in governance, if it is too low, then there's governance failure. So it is a very important factor in terms of, as you can see there, in terms of now influencing conservation. Also finance was another important factor, as you can notice there. However, you notice that national capital has a negative and it had a negative and significant influence on the conservation of lions, because theoretical, that is in fact an expected sign theoretically, because sometimes when natural resources are abundant, then communities do not see the need to cooperate or to collaborate and conserve the natural resources. And of course, when in any eventuality, like when there are so many bushes, then lions can also attack livestock. So they are found unawares. Again, also if they are too scarce, then there's no need of collaboration again. And also there's a lot of conflicts and also a lot of governance failure. So that is the explanation.
2 minutes, please.
Okay, I'm almost. So the mediation effect was also there between now the finance, congruence, and the land conservation. And also, there was also a mediation effect on most of these factors between them, like finance, through linking capital, congruence, and land conservation. Linking capital was they are the organizations at the ground level, like the NGOs, at the local level, and their importance in linking the local communities to higher organization levels, like the national level and the international level, where they get the funding to enable compensation. And that is why the mediation effect or the partial mediation was discovered, whereby each of these variables, like finance or any other variables like rules, monitoring, the direct effect between that variable and the outcome variable, the dependent variable, was weaker than the mediation effect. So finance was very, very important. Other important mediation pathways were discourse through linking capital and congruence, showing the importance of where the consensus between the discourse of different actors in conservation. So the policy recommendation is that an enhanced participatory governance should be enabled, whereby communities make rules together. There is monitoring that is participatory, because we found that also compliance had a negative effect on land conservation, because this kind of compliance is mostly coerced, and people, although they think there is, maybe they can attach that to success, they still have negative attitudes. So there should be enhancement of participatory governance, and inclusion of more and more actors, because one of the findings of this research is that in one of the group branches that we studied, it had the highest positive perceptions on lion conservation, and it is because it had a larger diversity of actors, more NGOs involved in it, and therefore they had more positive perceptions on lion conservation. Also, habitat conservation should be enhanced for lion conservation and community livelihoods. Thank you very much for listening.
Wonderful. Thank you very much for presenting. Just occurring to me, presenters are basically all from either University of Embu or now, is it Karatina? Ah, okay. None from our university here. I guess it's just a coincidence. Is it? Okay, so I believe we have also taken notes. Any questions or any reactions? So the very last one is a virtual presentation, which appears on our programs there. The last one on that page. Okay. Yes, this one is the presenter is in Japan, but I'm just guessing, looking at the authors there. He'll tell us. And maybe as he gets online, we can start. We can already react to one, at least Dr. Joan's presentation as this one comes on. So, remember Dr. John presenting about microplastics? Oh yes, we have one here. Microphone. Okay.
Hi, thank you very much for your interesting presentation. My simple conclusion was the level of microplastics in chicken dung was highest because they live closest to people. Because if I see the films and the other shapes, those were linked to what we consume as people. Isn't it? The bad thing is that. But you came up with only one recommendation, is to have this quality control of dung. But why not also trying to change the behavior of people so that they don't throw away and that they are more careful also with the waste?
Let me request the three presenters to come forward, please, so that because of logistics of microphone, I'll give you this one of mine so that to reserve that, so that we use this one, so that it's faster. Okay. Maybe we can take all for John. Any other?
Yeah.
So that we actually react.
I have already been Now, I was also quite triggered by your presentation, so thank you very much. Very nice. Pity you didn't look for goat manure, because goats also have they're also eating everything. That's what farmers normally say, they're eating everything. So that is one, but it is indeed quite alarming. It is quite logic what you presented, but on the other side it's quite alarming. I thought we now have to worry about eating fish products. But what you are saying, animal products, it's going to be the same, and even plant products, because that's where you started. So I think indeed an important topic, but I totally agree with my colleague. We have to do something as people.
Thank you. Let me start with the coach. I hope I got your name right. Well, the recommendations as of now, because this is at a local level, is just to create that awareness that see, as much as you're going to use the compost manure that you're generating from the animal waste, it is a conduit or a channel to transfer microplastics. So very true that it's not abnormal that the chicken had the highest. It's because of its scavenging nature. It eats, almost picks on everything. And of course, yes. We also looked at the goat, and goat came second, and that could almost tell on the nature of the feed for the goats. And also from the farms that we collected these manure from, most of them give their animals supplements. I mean, extra feeds that are processed. So like for the cow, there's the dairy meal. For the chicken, there's also the chicken feed and all this. So part of this could be a contributory factor. So yes, I totally agree that if we are going to mitigate or control this, then one, we have to advocate for safe waste disposal or a better disposal. But on the other, that is on one, looking at it from one angle, I totally agree. On the other angle, in terms of the transfer to plants, which was mainly our focus, then that is why we were recommending the quality control. Yes, we looked at the goat also, and as I mentioned, it came second in terms of ranking: chicken, goat, and cow. So more or less, I would almost say, quote unquote, that if I were to recommend which of the three options would be most safe, then cow. Yes.
Thank you. But you can also more chicken than the Kinjiri chicken, isn't it?
Yes.
Thank you, Dr. for that presentation on microplastics. And now that gets me worried because we were going towards using of organic fertilizers as opposed to pesticides. Now what you're telling us is that even that small sham behind my home is not safe anymore. Now, what do you think? Is it not the high time now with such concern? Because we know these the fertilizers, the ones we usually use, usually go through, they actually analyzed by the quality. Now, is it the high time now, even the organic fertilizer to be controlled? Because now it is concerned from where they're coming from, then we might now bringing up, poisoning ourselves. Thank you.
Sure. Actually, this is a wake-up call that we can no longer treat our compost manure just like that and say that since it is organic, it is safe. So yes, I totally agree that then we need to start a conversation about how do we compost? What do we feed our animals on so that we may be able to use their waste and compost it into manure? So this is actually a start of a conversation that ideally, yes, even though we are moving towards the organic manure, it might not be as safe as compared to the synthetic fertilizers. So I totally agree.
Wonderful. Let's take the last one for John. Just the last one. Briefly, a brief answer as well, so that you go to the next person. There is someone waiting.
Thank you.
Yes.
Thank you very much, Dr. John Chede Ogule, for the wonderful presentation. My concern is that this is organic manure. I'm wondering, are we even safe, even with the inorganic ones? Because I look at the fertilizers, all the fertilizers that we have, their packaging inside is polythene. Basically, some of these fertilizers are microscopic in nature, corrosive, and I don't know what they do to the plastics. So, are we even safe with the inorganic type of fertilizers? Thank you.
Well, by the nature of the inorganic fertilizer, by its synthetic process, of course, it's not safe. Now, the packaging compounds the issue. It compounds the issue, and also the formulations that are used in some of these fertilizers. Remember, currently we are having what we call the slow release. So it's basically the fertilizer is packaged in some sort of a capsule. So what is this capsule? You see, so ideally, in terms of safety, the synthetic are worse. The synthetic are worse, and that is why the lately there's been this call that can we go organic, but still, even with organic, there needs to be some quality control. Thank you.
Thank you. Thank you very much. We go to Dr. here. John, who presented on extraction, CO2 extraction and all that. And we have any questions for him? To extract the oil, to purify the beeswax, and so on. And talking of the application, we heard today about avocados, and you know avocado oil is one big way of value addition matters avocado. Can this technique or technology come in handy, maybe in that respect, and would it be affordable, cheap, applicable, maybe compared to the existing ones?
Thank you. Yes, we were using carbon dioxide as the solvent. The way we usually do solvent extraction, and we use something like ethanol, hexane, all those organic solvents. Now we were substituting those organic solvents, and now we are using carbon dioxide, but in which region, not as a gas, but in supercritical region as a solvent. And yes, it's very applicable because when it comes to extraction or avocado, value addition, I'm seeing a lot of avocados now, even they're making those oils. And you can be able to extract the oil using this solvent. And the advantage of using this carbon dioxide, supercritical carbon dioxide, is that now, because the organic solvents are toxic, and the traces might be found if you extract using those solvents, then you might find those traces in that oil. And this oil will be used for human consumption. But when you use carbon dioxide, when it's released, then when you reduce the pressure and temperature, then you go back to gaseous after extracting your oil. Then you don't contaminate your oil. Then yes, it can be used. Thank you.
Wonderful, wonderful. Any other question for John? Yes, I can see two here. Let's have them. Let's hear them.
So, thank you, John. That was a very good contribution. I just want to hear from you, because now you are really emphasizing on the disadvantages of using organic extraction, and you are facing that with coating CO2. Now, we know organic solvents we select them depending on polarity and that which you want to extract. What about now carbon dioxide? Will it be limited by the polarity or the compounds or the fatal compounds maybe that I want to get from these issues?
Yes, thank you very much. That's true, that carbon dioxide is usually non-polar. So if you know the chemistry behind lights and lights, so you'll be extracting more of non-polar, okay? But now, the way we usually select now, maybe because we want to reduce more of usage of this organic solvent. So, as we know how we select it according to solubility or even toxicity. When it comes to green chemistry, it's reduction. So, we can modify the carbon dioxide. It has been done that use the supercritical carbon dioxide, but now you bring up something to modify, a modifier. Then you can mix the two, and then you can now be able to extract even non-polar components. Yes, but by using a less toxic organic solvent, and also the volume which will be used is reduced. Thank you.
Okay, let's have that as the last one.
Sorry. I was a little bit triggered by the statement that CO2 is a non-toxic. If I have too much in my blood, then something will happen. And I also was thinking about the relation, because CO2 is still a harmful greenhouse gas. So how do you relate your research to that?
CO2. So where, let's compare it to organic solvent. Organic solvents is petrochemicals. And of course, they are toxic. When we talk about CO2, okay, it's debatable. Is it toxic when you take the meaning of the toxicity? Okay, we usually breathe CO2. It's not toxic in our bodies, but again, we are not using it. We are not consuming that CO2. And two, it's greenhouse gases. Actually, we are using these rather than now from byproduct. When you are producing metal, you produce a lot of CO2. If you release it, then it becomes unfriendly to our environment. But what about we use this CO2 rather than releasing it to the environment? We package it and use this as a solvent. So that's the aspect. So the CO2 actually now with the green chemistry is that we are using those gases which are not very friendly, environmental friendly, and then we are trying to recycle them rather than now letting them to go to the environment.
Wonderful. Thank you very much, Ajahn. We have the seats. We have this other DART presentation, reactions to the DART presentation. To Margaret? Yes, to Margaret. Conservation of lions in Abosel, using CBC approach. At CBC, nothing to do with the education system. So conservation, right? Okay, there's one here.
I want to thank all for the good presentations. I have one concern to Margaret. You talked about using mixed methods in your methodology, where you mentioned both qualitative and quantitative methods. Did that not make your research so massive? Were you able to do both in time? Thank you.
I would say yes, it was a very massive research, and I was able to do both, and I've not really reported so much on the qualitative data, but I was able to do both. For example, part of the qualitative data that we saw was on the discourse, the discourse that people had there on conservation, because mostly we noticed that the NGOs, the community leaders, and the Kenya Wildlife Service, they can coexist, they can live together. But as per the community, to them it is lions aside and them on the other side. There was no coexistence because there was so much cost to them and very little benefits coming from lions, from tourism, employment, or anything like that. So for them, they really believed there was no coexistence. They should be locked up in the Amboseli National Park to avoid them killing their livestock. But the others, they believed, because to them there was no direct cost, and they really believed that what they are doing is really working. They believe coexistence can happen. That was part of the qualitative data that I collected. So I can actually, I did them. I managed, but of course it was huge.
Okay. Any other question for Margaret? So why did you go to Aboseli, not Masai Mara, all the way from Nyeri to the border in Tanzania?
Aboseli has had a history of conflict. And if you look at Masai Mara, of course they also have conflict, but Masai Mara, the governance there, in fact it is normally compared. If I had adequate money, I would have collected data in the two areas and compared the data. So the history of Mara, the governance has not been very bad as Aboseli. There have been a lot of now governance issues in the Aboseli, and in fact currently the Aboseli National Park is about to be handed down to the county government of Kajiado. I hope they will manage, but it's one way of devolving governance, which should have happened from the beginning. Yeah.
Thank you. Very good answers. Thank you. I think we can let you have your seat. We have one? Okay. Okay.
Yes, thank you for your interesting presentation. You talk about community. Now, I'm also wondering, do men, women, youngsters, elders are equally impacted by the Lions? And how are they represented in this community-based governance system?
That's a very good question, because I think that is an area for future research, because I noticed that the women may be impacted differently. It was outside the scope of this research, because women in the Maasai, they are left with the goats and sheep are normally like considered women property, and these ones may be predated more even by other predators other than the lions, so they might have a higher impact. And I still noticed that women have a responsibility of also feeding the home. So when one is killed, then they are impacted higher than men. Also, when you collect data in those areas, then you notice the participation of women in research is very much reduced. Because mostly they don't even want to talk, men want to give all the information. So I still tried to balance, particularly in the FGDs, by having groups only for women. Yes.
And if we look at the governance system, are women also represented there?
Absolutely no. Very few are represented. In fact, I could attend like meeting forums just for observation, and you got like maybe 5% of women attendance, and in some they were not there at all. So even in leadership positions, like the group branch leaders, then women are not there. For example, there is one NGO that deals with monitoring lions using indigenous knowledge, and they use the Morans. They convert Morans who are initially killers of lions to protectors of lions. But you see, these Morans, for them to kill lions, sorry, for them to kill lions, they are influenced by women also. There's a big influence from the women as well. So they should also be involved, but they are really like left out. Yeah.
Wow, thank you very much. Thank you. When you go back to Argentina, take our regards. Those from Embu, the same, I think they supported God. Why we don't leave, the conference is still on till tomorrow. All right. So thank you, audience, for your patience. So we usher in our last presenter. Hello, Bwana Kimathi. Can you hear us? Yeah, we can hear you. Maybe to use a mic for him. Okay. Yeah, we are putting you up. We hope you are ready. How is Utsunomiya? Is it morning or afternoon? Evening or night?
I've been waiting patiently.
Sorry we get you, but finally we are ready for you. So
The sequences, we selected a few sequences depending on the percentage similarity of the proteins. This virus protein has 6 genomes, and in this section, there has been cases reported of recombination, whereby a virus is formed by 2 viruses contributing genome to form a new virus that could be little than the parent viruses. In this case, the C4, C1 region has been very common. So from this table, you can see the tomato leaf curve virus, originally the one that was identified in Namibia, contributes the highest part in this case, apart from the replication gene and the C4 gene, the one responsible for symptoms determinant. However, in the tomato leaf curve, Uganda virus is contributing the 2 genes. So this one suggests a possibility of recombination, and therefore we checked the recombination. As I have explained, recombination in viruses is whereby viruses contribute sections of the genome to create a new virus. For instance, in this case, the tomato leaf curve Uganda virus contributed the C4 and the C1 genome, and the tomato leaf curve Bunene virus contributed the rest of the part. Now to form this new recombinant isolate that we identified from Portugal in Kenya. And when I compared to confirm the infectivity and whether this virus can infect any other plant apart from bottle we constructed an infectious clone and inoculated the clone to healthy plants, that is to healthy tomato, healthy cucumber, bottle which is the original host, and Nicotiana betamiana, of course the model plant. And as you can see from these figures, the symptoms are showing clearly, and they are similar to what was observed in the field. And this analysis can fulfill the coach postulate, which is very important as far as the infectivity and host range analysis is concerned. So, in conclusion, I can report that this is the first case of a tomato leaf curve virus infecting cucumber beet plants in Kenya. And from this, you can see the host range is quite diverse, and it warrants more need for more research to be able to determine its spread and how much loss it's causing to farmers. So this isolate may have occurred as a result of the combination, as I have explained earlier. So this is a research in progress, and so far from the good protein of this virus, we have been able, using the bioinformatics tools without purifying the virus, to select the epitopes that have been injected in rabbit and the
that we have on the screen. So, so how many accessions did you use here so that you come up with these so-called Kenyan isolates?
I used about 35 accessions, but I singled some out to at least to be left with members representing these regions described in the map.
Okay.
And also the one that showed a high percentage similarity with the identified isolate.
Okay. And so these are the percentage similarities or something? Or
Yes, percentage similarities, of the entire genome.
Okay.
Yeah, and maybe I can explain the comment you made on why
yes, please.
It's called the coronavirus.
Yes.
Actually, in the criteria for naming the viruses, the isolate we identified was 92.7% similar to atomatoviral coronavirus, so it could not pass the criteria for naming it as a new virus. So it has to be named it is a species of tomatoviral coronavirus occurring in Kenya. If it could be, for example, 87%, maybe we could have named it tomatoviral male virus or any other name, depending on where it has been collected.
Oh, okay. I'm well advised there. Yeah, because of course nomenclature follows certain rules and you cannot go against them. Now, the other question, I don't know how much, of course I know you're into virology more than entomology, but I was thinking about the vector, the Misia tabas, the white fly that you showed there, the vector. And wondering, because it's a very complicated species, it's a cryptic species with subtypes, genotypes, so to speak. So I don't know, have you been able to pin down which particular genotype of the vector is actually responsible for transmission of this new virus? Maybe even from region.
During my PhD study, I have not confirmed, but when I was in the same university for masters, and I confirmed some white flies we had collected in 2017 from tomatoes that were infected with the virus, the tomato leaf virus, and the biotype of white fly was called the Mediterranean group.
Okay.
Was the one that we identified. However, in this case, we did not collect white flies, so I cannot confidently confirm which biotype they belong to. However, in East Africa, the Mediterranean group dominated.
I see. So is it transmitted horizontally, vertically in the vector, or both, the virus?
Mostly it's by the vector, although there are some few cases where the transmission can also be passed via the seed.
Okay.
And I suspect there could be a seed transmission in this case because this both where the seeds are shared from one farmer to another, so I think it also warrants a better study to clarify the mode of transmission in this case.
Okay. Okay, I don't know, I don't see any other hand. Any other question from the audience? Looks like the audience has heard you and does not have any other issues to raise. So once again, I think you're doing beautiful work. Please keep on. Did you say you are in the same university for masters? Utsunomiya University?
Yes, I was in Utsunomiya University for masters, but now I am a PhD student at Tokyo University of Agriculture and Technology, but still based in Utsunomiya University. My research is at Utsunomiya University.
Okay. That's wonderful. Your bachelor's?
Probably I can mention that this research has also been part of the collaboration the UUA program with Dr. Cynthia Mugo and Professor Peka of Jomo Kenyatta University. So they have also been very influential in this study.
Oh, that's wonderful. Yeah, Dr. Cynthia is not in this conference today. She is bereaved. I think the funeral is on today. And so we give her our condolences. So I think with those many remarks, those few remarks, thank you very much, Bwana Kimani.